Now we will begin Session 3, Conquer Non-Communicable Disease by Control of Inflammation. I'd like to introduce our session chair, Dr. NANGAKU Masaomi from the University of Tokyo Hospital, who will be guiding us through this session. Our speaker for this session is Dr. Richard Kitching from Monash University and Monash Health. Dr. NANGAKU, over to you.
Thank you. First of all, I'd like to thank Professor Hirano and Professor Mishima for giving us this important opportunity to present our project here today. So our aim is to conquer non-communicable disease such as diabetes, cardiovascular disease, and kidney disease via control of inflammation. We have the inflammation-centric view about this, and we are going to achieve the healthy longevity by innovative telemedicine, accessible anywhere. We are discovering novel inflammatory cytokines that can be utilized to control chronic inflammation at home. To control chronic inflammation at home, we need to develop remote sensing technology, and we need new methodology to control inflammation at home via electrical stimulation, magnetic stimulation, and exercise mimicking drug. Many factors can contribute to chronic inflammation, such as diet, stress, and sleep disturbances. And to implement our findings to the applications for our patients and people, we need to develop new medical network. This will only be performed via team effort based on the strong collaboration between the field of medicine and the field of engineering. We are discovering new inflammatory cytokines which are secreted from the locomotor organs such as muscles and bones through exercise. Exercise is known to be good for health, and exercise can control chronic inflammation. And we are going to use these new cytokines as a new biomarker of exercise and eventually as a target to control inflammation at home. We are also exploring the possibility of individualized feedback of inflammation control by understanding the correlation between sleep and exercise in collaboration with a group from Sweden. And we are developing the innovative inflammation sensing technology. Skin gases are particularly attractive markers compared to exhalated breath, which can be measured unconsciously and continuously in a daily life. However, body gases are very low in concentration, and we need the breakthrough technology to sense body gas, skin gas. And actually, we have been successful in developing this new system. Our new system, electrical nodes, have high sensitivity as low as ppb levels. This is 3 orders more sensitive than the current best instrument. And we perform gas detection by using 10 nanometer size controlled oxide semiconductors. We are also establishing the method to control inflammation for evoking regulated inflammation. Through this, we are going to develop the new methods such as electrical stimulation, magnetic stimulation, and exercise mimicking drugs. We are currently using a model of sarcopenia by tail suspension, but somebody proposed that we should collaborate with JAXA and put these mice in the space so that to induce sarcopenia, and that is an interesting proposal. We are also developing the innovative technology for next generation diagnostic testing to bring hospital into home. To perform the future telemedicine, we need to measure the targeting molecules using only a small amount of substance. And we are developing the new generation digital biomassay by developing new molecular handling and detection technologies. This will enable us to use a palm top-sized system. We have succeeded in the enrichment of a factor of 100, and we can do this enrichment without the external instrument, external electric power supply. And to implement these, we need to develop new medical network, and we are going to use the HCell L7 Fire for healthcare services and connect the data at home and the data at hospitals. We have a good team for the statistics, and we have a good team for administration, and we are going to achieve this goal and achieve the healthy longevity by 2040. And as we are going to control inflammation, we need expertise about the inflammation. And we have a great collaborator, our good friend Richard Kitching from Monash Health Center for Inflammatory Disease, Monash Health. And I'd like to ask him to give his lecture. Richard, please.
Thank you, Professor NANGAKU. That's great. It is going good. Look, it's a great pleasure and a privilege to be invited to speak here. I'm very pleased to be here, and today I'll be talking largely about CD4 positive T cells in glomerular inflammation. These are my disclosures. Before I start talking about CD4 positive T cells, I do want to make a couple of remarks about chronic kidney disease and kidney disease worldwide. And I'd like to start by saying that Japan has the highest prevalence of known chronic kidney disease in the world at 20.2%. And these are data generated by Professor NANGAKU and colleagues and published in the most recent International Society of Nephrology Global Health Kidney Atlas in 2023. Japan, furthermore, has the second highest prevalence of treated end-stage kidney disease in the world. Those are people on dialysis or having received a renal transplant at 2,696 per million people, which is more than 1 in 500 people on renal replacement therapy currently in Japan. And clearly, this comes at a high cost, both personal and economic. I don't probably need to tell you, being inflammationologists yourselves, that common causes of kidney failure all have an inflammatory component. Obvious ones such as glomerulonephritis that I'll be speaking about today, but also perhaps less obvious ones including vascular and hypertensive disease, diabetes, genetic causes and acute kidney injury all have significant inflammatory components. And these are three pictures at low power of renal biopsy cause. The first one looking normal, the second one with the two arrows showing acute inflammatory changes, the top arrow being the glomerulus, the small organelle that I'll be discussing today within the kidney, and the bottom arrow relating to the tubulointerstitium. And what we try and do in acute inflammatory change is turn that around and resolve that inflammation before it leads to changes that you can see in the third panel. Here, the black dots are sclerosed and fibrosed glomeruli that will no longer work and perform their filtration activities. And the sort of empty grey bit is the tubules that have atrophied and been replaced by matrix proteins. So glomeruli are unusual capillary beds. They're high flow, high pressure filters with high shear stress. They are bounded by arterioles that also make them unusual. And as such, they're vulnerable to a wide range of inflammatory components, including in situ and circulating immune complexes and their related activation of complement and innate leukocytes, but also T-cell mediated injury. And furthermore, as I've mentioned, even things that are supposedly non-inflammatory have a significant inflammatory component, including genetic disorders, hypertension, diabetes, certainly preeclampsia, and disorders of complement. So this is by no means specific to the kidney, and I'm sure you're all aware of these sort of concepts. But what we tend to do in kidney disease is try and do a number of things. Switch off local inflammation and block chronic inflammation is fairly obvious. this hopefully leads to inducing healing and repair in the kidney rather than progressive fibrosis. And there have been developments in the progressive fibrosis field of drugs that were initially thought to be not related to inflammation, but also have in fact have a significant anti-inflammatory effect. But it's probably not enough to do that just alone. The underlying drivers of disease in the kidney are often, in fact, usually outside of the kidney, whether they're immune or not, and it's important to try and switch off and remove these underlying drivers of disease and prevent further effector responses. So I'm going to speak about glomerulonephritis, and I'm going to focus on autoimmune glomerulonephritis, but I would preface that by saying that I think in the glomerulonephritis field, we've become a bit disconnected from immunology and inflammation, in part because of the nomenclature we use. This is based on renal biopsy descriptions and histological lesions rather than cause, and my colleagues and I recently have written a couple of articles proposing a different categorization of glomerulonephritis, quite simply according to the cause. Nephritis can be related to infection or the immune response to infection, to autoimmunity, occurring in the context of transplantation. It can be due to autoinflammatory diseases, and lastly, due to monoclonal gammopathies. And I'll be talking today exclusively about autoimmune glomerulonephritis. Specifically, the most feared type of glomerulonephritis, that is to say, rapidly progressive glomerulonephritis. These are lesions that develop quickly over a period of days to weeks, causing renal failure from weeks to months. And I'll be talking about three elements in this talk. Firstly, how Class 2, HLA Class 2, which we've heard about previously this morning, predisposes to and protects from disease. And then the last two shorter parts are linked, really looking at effector responses in the kidney, how antibodies and T cells cooperate in an unholy alliance to affect injury, and how antigen is recognized in the glomerulus. So we've studied Goodpasture disease, or antiglomerular basement membrane disease, as a model autoimmune disease in a reductionist manner, and it's got several characteristics that allow us to do that. Firstly, the autoantigen is clearly defined as the non-collagenous domain of the alpha-3 chain of type 4 collagen. Now, as you know, collagens are matrix proteins, and type 4 collagens are involved in basement membranes, forming a triple helix. And specifically, the alpha-3, 4, 5 chains are involved in specialized basement membranes, particularly in the glomerulus and the lung. We're lucky, and these are highly conserved, so there's a strong human-mouse homology. So that's one reason why we can move between mouse and human in our observations and experimental studies. This disease has a fairly gold standard diagnosis. People don't turn up. People turn up with the disease. The disease is diagnosed effectively and accurately, and there's little in the way of overlap. They have standard autoantibodies that are almost always measurable in the serum, and on kidney biopsy, you can see rapid progressive glomerulonephritis with destructive changes in the glomerulus, together with linear IgG on the basement membrane reflecting its binding to the autoantigen. And lastly, there are strong HLA associations, this time not with DQ, but with DR. And I probably didn't need to make this slide, but I have. This is the concept of Class 2 setting the scene, or setting the stage for the peeling away of the layers of tolerance that occurs in the development of most autoimmune diseases. And you can see that most autoimmune diseases are associated with a risk HLA, but many of them, including diabetes mellitus, as we heard today, have a negative or modifier HLA. And Goodpasture disease is one of these. There is a strong positive association with HLA DR15. Unlike diabetes, it doesn't seem to make much difference in most situations whether you have one copy of that gene or two copies of that gene. The risk is there. There are negatively associated HLAs, and the one I'm going to talk about today is HLA DR1. And I said it doesn't matter what you have as the other gene, except if you have DR15 and DR1, where epidemiologically you're protected from the risk of disease. In other words, DR15 predisposes to the disease, DR1 in a dominant manner protects from disease. And so we used HLA humanized mice and humans, of course, to define the biology of this disease. And these mice have no mouse class 2, and their peptide binding domain in class 2 is encoded by the non-polymorphic DR alpha 1 chain and the highly polymorphic DR beta 1 chain, either DR15 or DR1. And therefore, one can use mice that have a homozygous for DR15, homozygous for DR1, or if you intercross them, they are heterozygous for DR15 and DR1, expressing about 50% of each. Therefore, their CD4 positive T cell repertoire is largely defined by the interactions between DR and the endogenous peptides in the mouse. And in good pasture disease, when we started this work, there was a limited amount of information about epitopes in this disease, but there were two key autoantibody epitopes, and there were two areas of T cell reactivity in a small human study. And we spent a fair bit of time in mouse and humans defining the immunodominant and disease-initiating peptide in this condition. And it narrows down to a minimum of 11 amino acids, alpha-3, 135 to 145, with strong homology between mouse and humans. And I'll get this out of the way right at the start. We did most of our studies in mouse and in human peptides to ensure that what we were observing was not a relationship of a reaction to altered self. So similar findings in both the mouse and the human peptide when we worked in the mouse. So we did a number of studies, and just to summarize them here to show that this epitope was pathogenic in the context of DR15. Firstly, humans with disease compared to fully HLA-type normals had 100-fold enrichment of these alpha-3, 135 to 145 specific T cells measured by HLA class 2 DR15 alpha-3, 135 to 145 tetramers. These were largely, as one expected, effector cells. In other words, they were FOXP3 negative. In a mouse model of this disease, if you take kidneys and you dissect them out, you can see, again, the accumulation of tetramer-specific T cells that are largely FOXP3 negative. This is not a spontaneous model. You have to immunize the mice with either peptide or with whole antigen, importantly. It induces autoimmunity, T cell responses, autoantibodies, and disease. And it does this in DR15 transgenic mice, but not in DR1 transgenic mice, and also not in DR15, DR1 double transgenic mice. In other words, this recapitulates the epidemiological observations about this disease. We made T cell clones, CD4 positive T cell clones, and this could transfer disease into naive DR15 positive mice. And using a series of peptide and antigen screening studies across the full length of the non-collagenous domain of the alpha-3 chain of type 4 collagen, we were able to show that the pro-inflammatory T cell reactivity between DR15, DR1, and double transgenic DR15, DR1 mice differed only at alpha-3, 136 to 146. In other words, this was the peptide that made these mice different to resistant mice. So we went to look at the structural basis of the interactions of this peptide with DR15 and DR1. And in collaboration with Professor Jamie Rossjohn at Monash University and Hugh Reid, we found something that they thought was very interesting, and I tended to agree with them because they were the experts. It's what's called a register shift. So in other words, the peptide could bind both DR15 and DR1, but it moved along one. In other words, if you look at pocket 1 here, it's highlighted by a red box. There's an isoleucine in the DR15 bound to pocket 1, and there's a tryptophan bound in the DR1. This results in a backbone flip in the peptide, so it's now facing a different way. And we believe and contend this would lead to different T cell contacts when undergoing selection. And now I'm going to try and make this video work. And I can't, which is interesting. Please. That needs to work. There's a number of videos in this. Yeah, it's a video. Okay, I'll take the pointer off. That's a better idea. Okay, so now we're looking down into the DR15 and DR1. The yellow is DR1. Blue, so that's not true. That is true. Blue is DR15. We're looking down. There are 9 amino acid difference between the two, large in the binding pocket. Peptide pockets 1, 6, and 7 are very capacious in DR15, allowing the bulky aromatic side chains to bind and sit down in the pocket. So you can see this wide area here. If you look at DR1, there's an alanine cutting off that's separating those pockets, and they can no longer bind in this way, but pocket 1 is deeper. The backbone flips. The peptide is now bound down, but with some of the aromatic side chains from the phenylalanine tryptophan poking upwards, allowing potentially T-cell contact and potentially affecting T-cell selection. So we wanted to ask a couple of questions. Why is DR1 protective, obviously, and how does DR1 afford this dominant protection from the DR15 associated risk of disease? And in data I won't be able to show you today for time, we showed that the naive repertoire in humans and in mice was different, the naive antigen-specific repertoire in terms of T-cell receptor usage was different between DR15 and DR1 mice and humans. We wanted to look at the impact this had on the antigen-specific phenotype. And this is a slightly complicated slide, but we've done this in mice, and I'll show you in humans in a minute. The gist is in normal mice that are DR15 positive, the tetramer positive cells are largely FOXP3 naive effectors. However, in DR1 transgenic mice, the T cells that are antigen specific are largely FOXP3 positive regulatory cells. So DR15 and DR15-1 generates naive effectors predominantly that are specific for this disease-initiating peptide. In DR1, you generate regulatory cells, and it holds true across when you look at the double transgenic mice. What happens in humans? Well, these are fully HLA-typed DR15 homozygotes, DR1 homozygotes, or DR15 and DR1 heterozygotes, and you can see without going through all the data in fine detail, the pattern is the same. DR15 tends to select naive effectors, DR1 tends to select regulatory cells. So the hypothesis that we've been working on, is that FOXP3 negative cells in the other things that can happen in the development of autoimmunity set the tone for the development of follicular helper cells or T-cell effectors, but in the presence of DR1, the regulatory cells that are antigen-specific can protect from the risk of autoimmunity and disease. And we further this by looking at regulatory cell depletion in vitro and in vivo. So here we used an existing technique to induce what is really a primary immune response over 8 days to this peptide. So we took T cells from fully HLA-typed humans and we put them in culture, either with or without regulatory T cells sorted out to start off with. And these were the results. The open bars are the Treg intact cultures from either DR15, DR1, and DR15, DR1. The closed bars are the cultures results in which the regulatory cells have been removed, and you can see that only the DR15 positive humans were able to break tolerance to this peptide and produce pro-inflammatory cytokines. However, there was breaking of tolerance in the double transgenic mice. In the heterozygous humans, when regulatory cells were depleted. So this shows that regulatory cells are suppressing and maintaining tolerance in these cultures. Furthermore, we could measure antigen-specific IL-10 and TGF-beta production that occurred only in the context of DR1 when there was regulatory cells present. And we did a similar thing using mice, showing that these antigen-specific Tregs are highly potent in preventing loss of tolerance in vitro. So the last experiment we wanted to do was to take this to an in vivo model of disease, and therefore we immunized repeatedly with peptides DR15 transgenic mice, DR1 transgenic mice, or mice that were both transgenic for DR1 and DR15. And in naive mice, if you use an anti-CD25 monoclonal antibody, you can deplete regulatory cells for around about 17 to 20 days. So we did this. And you can see the results are largely similar to what we saw in the in vitro studies. That is to say, only the DR15 transgenic mice developed disease in the presence of regulatory cells, but when regulatory cells were depleted, the mice that bore both DR15 and DR1 developed histological and functional evidence of disease together with autoantibodies that are characteristics of this disease in humans. So just to summarize, in the bottom line here, if you are DR15 positive, you have the capacity to generate largish numbers of naive potential effector cells to this disease-initiating peptide. If you are unlucky and go through a number of other steps, including antigen exposure, environmental factors, and infection, you may develop this disease. Unless you have DR1, where your large numbers of antigen-specific regulatory cells protect you from this process. So we've found that HLA polymorphisms shape the relative abundance of these self-epitope-specific Tregs in mice and in humans, and we think that determines the underlying protection or susceptibility to autoimmunity that we observe in this disease. Clearly, antigen-specific T cells help maintain tolerance, and clearly they may be able to use to treat other autoimmune diseases. And it may, I would speculate, and here in the previous talk, it doesn't sound like it happens in diabetes, but it may help explain some of the aspects of HLA-mediated protection susceptibility in other autoimmune diseases. So I want to spend the rest of my time in the talk talking about effector responses in the kidney. So loss of tolerance is one thing, effector responses is also important. And I'm going to switch to another disease here. This is another form of rapidly progressive glomerulonephritis, that clinical and histological picture where one develops rapid destruction of glomeruli with rapid progression of renal failure over days to weeks. And the disease I'm going to be talking about in the last few minutes of the talk is ANCA-associated vasculitis. So this is a small vessel vasculitis that particularly and critically involves the kidney. One develops loss of tolerance to one, and usually only one, of the neutrophil proteins, proteinase 3 or myeloperoxidase, and I'll be speaking about myeloperoxidase or MPO specifically today. Similar events are thought to happen in PR3, but there's a lack of an animal model for PR3 for reasons that we don't need to discuss today. Anyway, you lose tolerance to myeloperoxidase, you develop T and B cell tolerance, you develop autoantibodies known as ANCA, anti-neutrophil cytoplasmic antibodies. And these antibodies bind to the neutrophil, and instead of killing the neutrophil, this is not a neutropenic disease, it activates the neutrophil, they activate the neutrophils then lodge in an active process in vulnerable vascular beds, particularly in the glomerulus, where they induce microvascular injury, but also deposit their autoantigen. And this means the autoantigen is available and potentially for recognition by affected T cells. And although human pathologists in human disease don't routinely stain for CD4 positive T cells in kidney biopsies, there is a powerful rationale for the involvement of these T cells in rapidly progressive glomerulonephritis. Firstly, as I've shown you in the previous section of the talk, you can find autoantigen-specific CD4 positive T cells in humans and animal models. These antigens can be endogenous, such as the antigen I was talking about, the alpha-3,4-NC1, which is found in the glomerular basement membrane itself, or it can be lodged or planted in glomeruli, as I'm about to show you. CD4 positive T cells are found in glomeruli in patients with this syndrome, and experimentally we and others have shown in model systems and in disease models themselves that antigens in glomeruli can be recognized by affected T cells to mediate glomerulonephritis experimentally. And so I wanted to show you this in human studies that we performed. MPO, myeloperoxidase or MPO here is in red. It's not particularly important that we spend some time discussing nets, but nets are formed in this condition. These are human glomeruli, and you can see degranulation in the top panel, some other form of neutrophil-mediated cell death, or neutrophil cell death, which is probably necroptosis or apoptosis, but we haven't taken that further, or netosis, all occurring in glomeruli. But perhaps the most important picture I want to show you today in this slide is the presence of leukocyte-free MPO, or myeloperoxidase, the autoantigen now in the kidney. So you can see that bright red pink blob is a neutrophil with MPO, but the arrowhead points to myeloperoxidase that is lying between the endothelial CD34 staining and the podocyte nephrin staining. So here the autoantigen has been lodged in the glomerulus, and there is plenty there. So we spent some time ago defining the immunodominant epitope, T cell epitope of myeloperoxidase, which has since been validated in human studies and is part of an epitope hotspot in this molecule. And we generated Th1 interferon gamma secreting CD4 positive T cell clones, and we transferred to this epitope, and we transferred them to rag knockout mice. And then we did what is considered to be the standard model of the disease, which is priming neutrophils with LPS and then passively transferring on anti-MPO antibodies. And at day 14, we study injury. So we first wanted to show that analogous to the humans, there is extra leukocyte MPO present in glomeruli, and the top panel shows neutrophil recruitment that occurs in the glomerulus. The middle panel shows the increase in MPO activity in the glomerulus after this procedure, and the bottom panel in green shows myeloperoxidase that is not associated with leukocytes, the red being CD45. And the controls, I should mention, were ovalbumin-specific T-cell clones, and at day 14, there was mild injury, the injury that was induced by the autoantibody and the neutrophil. But by day 14, there was a dose-dependent, quite severe glomerular injury. You can see the pink PAS-positive material there, showing segmental necrosis and glomerular fibrin deposition, which is a key manifestation of delayed type hypersensitivity, which itself is a manifestation of a Th1 response. And we did this several different ways. We did this by transferring anti-glomerular basement membrane antibodies in, the so-called nephrotoxic serum nephritis or Mizugi model, where neutrophils are recruited rapidly and then disperse. Having said that, they deposit myeloperoxidase in the glomerular microvasculature. We also did it in a proof of concept model. In the absence of other inflammation, we used an antibody called 8D1, a monoclonal antibody we have that's an IgG1 monoclonal antibody that binds to the glomerular basement membrane, but because it is a mouse IgG1, it does not engage Fc receptors or complement well at all, and thus does not induce significant injury in itself. So we planted the peptide in the kidney. And at each of these occasions, transfer of CD positive T-cell clones specific for myeloperoxidase induced acute and relatively severe injury with segmental necrosis and a degree of crescent formation. So we know that in this situation, T-cells can recognize the antigen in or around the glomerular capillary in quite an acute situation where there is not a lot of endothelial damage. The question is, how does this happen? I'm going to spend the last part of the talk talking about some data that we have published showing a unique mechanism of microvascular, intravascular antigen recognition by CD8 positive T cells. And this is some work I've done with my colleague Michael Hickey over many years. On the left, there is a normal human kidney biopsy of a slice stained for monocyte macrophages, and there are none there. In disease, they are there. And so this was the dominant paradigm at the time. But using dual photon intravital microscopy of the kidney, this is a close-up of single glomerulus with a fractalkine CX3CR1 GFP mouse, monocyte from that patrolling within the glomerulus. And if you quantitate that, you can see over an hour, each glomerulus hosts around 8 monocytes that crawl around for around 15 to 20 minutes. Most of them are crawling. They crawl at 6 microns per minute. So these are monocytes that happens in other elements of the microvascular, for example, postcapillary venules, but these are monocytes that are patrolling the glomerular capillaries. So to go and ask the question of whether these monocytes could be relevant to T cells, we turned to a model that I established some years ago, again in a reductionist model, using a model antigen to see how T cells could induce disease. And here, the monoclonal antibody I mentioned before, the 8D1 monoclonal antibody that's directed against the glomerular basement membrane, but is a relatively non-nephrotic antibody. You can see it binding here in turquoise in the glomerular basement membrane, is coupled not to myeloperoxidase, but to ovalbumin, or the immunodominant ovalbumin peptide. So in this way, without inducing a lot of injury or any measurable injury at all, in fact, we can plant an antigen in the kidney. We take naive OT2 cells. This is a standard transgenic TCR transgenic line, specific for ovalbumin 323 to 339. We culture them up. You can generate Th1 effector cells fairly easily this way. We label them fluorescently, and we transfer them to the mouse. And if you transfer in the over peptide coupled antibody, you can therefore have an antigen that's planted relatively specifically within the kidney. So we want to address the hypothesis that the monocyte was actually presenting the antigen to the T cell within the glomerular capillary. And let's have another go at one of these videos. And this is an OT2 cell with antigen, hadn't stained for the antigen, crawling around in the glomerulus and spending about half an hour in the glomerulus. So we do know that affected T cells in the system crawl around within the glomerulus. What are the options for antigen presentation here? Well, there are 4. Endothelial cells, dendritic cells, B cells, or monocytes. Dendritic cells are extremely uncommon in the glomerulus, and only in severe disease do they come in any numbers. We did some functional studies to exclude DCs. Naive B cells are not particularly good at presenting antigen. We also did functional studies to exclude B cells that I won't have time to show you here. We did, however, look at class 2, and these are, okay, yep, these are, yeah, it's sort of working. These are MHC class 2 transgenic mice. Sorry, MHC, not MHC class 2 transgenic mice at all. They're MHC GFP mice, so the major histocompatibility complex class 2 is fluorescing green, and you can see the blue was outlining the endothelium of the glomerulus, and there is no expression of class 2 by the glomerulus, by the glomerular endothelium. However, when you look at these mice, the class 2 GFP mice with labeled T cells, you can see prolonged contacts occurring within the vasculature of the glomerulus after the antigen is presented. And to cut a fairly long story short, we showed that these cells were CD11B positive monocytes. They're the ones that spent a long time in the glomerulus. I showed you that previously. So these monocytes are contacting CD4 positive T cells and have the capacity to present antigen to them. So we looked to see if this activation within the kidney was antigen dependent. If you inject, these mice all received T cells that were activated ex vivo, and the readout here was interferon gamma production by flow cytometry. Mice receiving 8D1 antibody alone did not have much in the way of activation. Mice receiving the ovalbumin peptide copied to an irrelevant immunoglobulin also did not have much activation. In other words, the antigen was present but not in the kidney. It's only when the antigen was present in the kidney that the T cells were activated, which is what we might expect and hope. We also looked at downstream effectors in terms of the accumulation of neutrophils and monocytes and found a similar thing. So we transfected the T cells with a GFP NFAT1, which moves the nucleus on TCR signaling to try and detect in vivo whether TCR signaling is occurring in this context. And you can see the ex vivo in vitro study there. You can see the NFAT in the cytoplasm, the nuclear and cytoplasm, and lastly in the nucleus. And when you don't inject antigen, these are the T cells in red, GFP in GFP, the NFAT in GFP and GFP. You can see that it remains cytoplasmic, so no T cell signaling of any note. Having said that, this is a ovalbumin-specific T cell in the kidney that has undergone T cell signaling from the monocyte that's presenting the antigen to it, and the NFAT has moved to the nucleus. And you can quantitate this, and we did. And we found in the presence of antigen, such transactions occurred more frequently. Now, luckily, it's not very effective. It's not a good way of recognizing antigen, and that's merciful. I don't think we would have evolved if that was the case. If every time we had an antigen that was present in or around blood vessels, we would then find that, that there'd be widespread vascular inflammation. But it can happen. It does happen in glomerulonephritis experimentally, and we believe this has relevance to humans. So just to summary, inflammation is critical to both acute and progressive kidney disease. Kidney disease is common and important and an increasing burden on society. But back to the CD4 positive T cells, autoantigen-specific regulatory T cells are preferentially generated by some class 2 allomorphs in humans and help maintain tolerance. I should mention something that I didn't. These humans and these mice have similar numbers of total regulatory cells. The changes are antigen-specific, not throughout the regulatory cell population. Turning to ANCA-associated glomerulonephritis, I think this is a unique effector response where we have humoral autoantibodies that instead of destroying a cell, sends that cell to a vulnerable and so-called innocent vascular bed, microvascular bed, where it induces injury, but also releases autoantigen, allowing this antigen to be recognized by effector CD4 positive T cells. So I guess it's a combined class 2 DTH-style hypersensitivity reaction. And lastly, this happens, at least in part, by CD4 positive T cells recognizing antigen presented in glomerular capillaries by patrolling monocytes. And I'd just like to thank my current research group, previous lab members who have worked on this work, my collaboration with Michael Hickey and Jamie Rossjohn at Monash, and collaborators in Oxford and Vanderbilt. Thank you, and I'm happy to take questions.
Thank you for the presentation. We'll now proceed to the Q&A session. Before we begin with the audience questions, we have questions from Dr. NANGAKU, specifically directed to Dr. Kitching. Dr. NANGAKU, please go ahead with your question.
Thank you, Richard, for your comprehensive and beautiful talk. To control the chronic inflammation in other non-communicable diseases, such as diabetes and cardiovascular disease, what kind of the strategy do you think the most appropriate?
Well, that's a very difficult question to start with. Look, I think, if I could phrase it this way, I think it would be, and we've thought about this for a number of years in inflammatory diseases, it would be nice if inflammation was similar in all different organs and tissues. But if I may paraphrase Dostoevsky, all happy families are unhappy in a different way. And I'd be concerned that what we're taking for the kidney may or may not work in other areas. It would be nice if it did, and I've gone through my career hoping this was the case. And perhaps with some drugs like the SGLT2 inhibitors, this might be the case. So it would be good to find a blockbuster drug that worked locally and systemically in many different organs. The downside of that, I guess, is if you are influencing protective immunity substantially, you may risk acceleration, for instance, of tumour growth that we heard about this morning. And it reminds me that I was talking to Professor NANGAKU that in 2001, all the talk about PD-1 and CTLA-4 was about enhancing it to prevent autoimmunity. It was only when the cancer biologists and the immunologists got together, and mercifully they did, that this was the idea of checkpoint inhibition was translated to clinical reality. So I think this yin and yang of the immune system, and both innate and adaptive, is a really important one to consider. And we can get carried away with blocking inflammation without thinking about what it does to protective immunity and protective inflammatory responses. I don't know if that's answered your question, but I've decided, I think, to answer a different question than the one you've asked. But I think that's a really important question, and we should all bear that in mind.
Thank you, Richard. And it is interesting that you mentioned SGLT2 inhibitor as the blockbuster drug. That's the drug which is now the first choice for cardiovascular disease and kidney disease. And the recent paper published in the Cell Metabolism this month showed that canagliflozin, one of the SGLT2 inhibitors, control T-cell function and may be a good candidate as a drug to autoimmune disease. So although based on clinical evidence, we know that SGLT2 inhibitors powerfully protects the heart and the kidney, actually we do not know the mechanism at this moment, and the mechanism may be through the control of inflammation.
And if I may add to it, I think time and time again we've found not so much cytokines, but molecules that are thought to have one narrow function, because that's what we discovered first, that actually have quite a broad action. And I think we need to keep an open mind on that, because some of the so-called off-target effects of this drug, for example, may be critical in its action.
Thank you.